Hello and welcome everyone. I am Song Lee. Today I would like to present about a DNA binding protein called MeCP2. Before introducing the protein, it would be great to have a quick overview on DNA methylation. DNA methylation is when a methyl group is added to the 5 positions of cytosines of DNA, and this modification represses gene activity. The enzymes responsible for the addition and maintenance of methylation markers are DNA methyltransferase proteins, also known as DNMT proteins. How does such a small modification to DNA exert its biological effect? In other words, who reads the DNA methylation markers? That is when our protein MeCP2 comes in. MeCP2 is the abbreviated name for methyl CpG binding protein 2. This protein was discovered for its affinity towards methylated CpGs. Its binding ability comes from the MBD, whose structure is shown on the right. The gene for this protein is located on the X chromosome. Over the years, numerous interacting proteins were identified. A figure by Tillotson and Burt wonderfully summarizes the interacting proteins. The blue bar indicates the location of MBD. Notice how many mutations which render MeCP2 inactive occur within the MBD. This shows that MBD is essential for the function of MeCP2. The big red box indicates the transcription repression domain, which interacts with various proteins that repress transcription. Shown here are the protein complexes formed by MeCP2. Early experiments showed MeCP2 binds transcription repressors HDAC and MSyn3A. HDAC, or histone deacetylases, represses gene expression. Thus, MeCP2 can be thought of as an important protein linking the two inhibitory epigenetic mechanisms, DNA methylation and histone deacetylation. More recent evidence reports MeCP2 is also able to activate gene transcription when binding to other epigenetic markers. If MeCP2 is such an important protein, what disease is associated with it? It was discovered that MeCP2 mutations are responsible for Rett syndromes. Rett syndrome is a neurological disorder, mostly occurring in female patients, attributable to the fact that MeCP2 is X-linked. Patients show severe autistic symptoms. However, it was demonstrated that reactivation of MeCP2 reverses Rett syndrome-like symptoms in mice models. This is a surprising result, and thus it was called the miracle mouse. These are the presentation notes, and thank you for your attention.